Hello everyone, I'm back with a new tutorial video on carbohydrate classification. I hope you get good information on this topic. Don't forget to subscribe to the channel and like the video. Let's continue with the introduction. Carbohydrates are usually defined as polyhydroxyaldehydes and ketones or substances that hydrolyze to yield polyhydroxyaldehydes and ketones. Simple carbohydrates are known as sugars or saccharide. Carbohydrates are classified based on their chemical structure and complexity. Here are the main categories. 1. Monosaccharides. These are the simplest form of carbohydrates, consisting of a single sugar molecule. Examples include glucose, fructose, and galactose. 2. Disaccharides. These carbohydrates are composed of two monosaccharide molecules linked together. Common disaccharides include sucrose, table sugar, lactose, milk sugar, and maltose. 3. Oligosaccharides. These consist of 3 to 10 monosaccharide units linked together. They are found in foods like beans, legumes, and some vegetables. Examples include raffinose and stachyose. 4. Polysaccharides. These are complex carbohydrates made up of many monosaccharide units. Understanding higher-level carbohydrates depends on understanding the properties of monosaccharides, as they are the building blocks of them. Monosaccharides are the simplest form of carbohydrates, consisting of a single sugar molecule. They're the building blocks for more complex carbohydrates and play a crucial role in energy production and metabolism. Monosaccharides can be classified based on the number of carbon atoms they contain and the type of carbonyl group they contain. A. Aldoses. Monosaccharides with an aldehyde group, CHO, at the end of the molecule. Examples include glucose and galactose. B. Ketoses. Monosaccharides with a ketone group, C equals O, within the carbon chain. An example is fructose. Properties and structural complexity of monosaccharides. 1. Chiral center. A chiral center in a carbohydrate is a carbon atom that has 4 different groups attached to it, making it asymmetrical. This asymmetry allows for the existence of different stereoisomers, which are molecules that have the same molecular formula but differ in the spatial arrangement of their atoms. The specific arrangement of atoms around chiral centers can significantly affect the biological activity of carbohydrates. For example, the D and L forms of glucose have different properties and roles in biological systems. The orientation of the hydroxy group that is most distant from the carbonyl carbon determines whether the sugar belongs to D or L sugars. When the hydroxy group on this carbon is on the right, the sugar is D isomers. When it is on the left, the sugar is L isomers. Most of sugars present in biological system are D sugars. 2. Anomers. Anomers are a type of stereoisomer found in monosaccharides that differ in configuration at the anomeric carbon, which is the carbon derived from the carbonyl carbon, the carbon with the double-bonded oxygen, in the open-chain form of the sugar. When monosaccharides form ring structures, the carbonyl group reacts with a hydroxyl group on the same molecule, creating a new chiral center at the anomeric carbon. This reaction can produce 2 different configurations, known as alpha and beta anomers. For example, in glucose, the ring structure can form in 2 ways. Alpha anomer. The hydroxyl group attached to the anomeric carbon is on the opposite side, trans, of the ring relative to the CH2OH group. Beta anomer. The hydroxyl group attached to the anomeric carbon is on the same side, cis, of the ring relative to the CH2OH group. These anomers can interconvert in solution through a process called mutarotation, where the ring opens to the linear form and then recluses to form either the alpha or beta anomer. This property is important for the biological functions and reactivity of monosaccharides. 3. Epimers. Epimers. A type of isomer where 2 monosaccharides differ in configuration around only one specific carbon atom. For example, glucose and galactose are epimers, differing at the 4th carbon atom. We have discussed a lot of important points regarding monosaccharides. Now, let's proceed to complex carbohydrates starting from disaccharides. Disaccharides, C12H22O11, are sugars composed of 2 monosaccharide units that are joined by a carbon-oxygen-carbon linkage known as a glycosidic linkage. The disaccharides differ from one another in their monosaccharide constituents. For example, glucose plus fructose for sucrose, glucose plus galactose for lactose. And in the specific type of glycosidic linkage connecting them, for example alpha or beta linkage, between the monosaccharides. One important point to consider in disaccharides is the glycosidic bond formation. Sucrose's glycosidic bond is unique because it links the anomeric carbon of glucose, alpha-D-glucose, to the anomeric carbon of fructose, beta-D-fructose, through an alpha-1,2 bond. This bond formation involves both monosaccharides' anomeric carbons, making sucrose a non-reducing sugar. In contrast, other disaccharides like lactose and maltose have glycosidic bonds involving only one anomeric carbon, leaving the other free, which makes them reducing sugars. The other complex carbohydrate is polysaccharide. Polysaccharides are complex carbohydrates composed of many monosaccharide units linked together. They serve various functions, such as energy storage and structural support. Common polysaccharides include starch, glycogen, and cellulose. Starch. Starch is a polysaccharide that serves as a primary energy storage molecule in plants. It is composed of two types of molecules, amylose and amylopectin. Amylose is a linear chain of glucose units linked by alpha-1,4 glycosidic bonds, while amylopectin is a branch molecule with both alpha-1,4 and alpha-1,6 glycosidic bonds. Glycogen. Glycogen is a polysaccharide that serves as the primary storage form of glucose in animals. It is highly branched and composed of glucose units linked by alpha-1,4 glycosidic bonds, with branches formed by alpha-1,6 glycosidic bonds. This structure allows for rapid release of glucose when energy is needed, making glycogen an efficient energy reserve. Cellulose. Cellulose is a polysaccharide that serves as a structural component in the cell walls of plants. It is composed of long chains of beta-D-glucose units linked by beta-1,4 glycosidic bonds. This unique bonding pattern gives cellulose its rigidity and strength, making it an essential material for plant structure. That's all for today. As always, I'll be back soon with a new topic. Thank you.